Hello again, welcome to another Show Me tutorial that will hopefully help you with your learning on the Foundations course. Now today we are going to look at the spinal cord in a little bit of detail, and in order to do that, first of all we're going to take a look at this diagram and label it. So let's have a look at what we've got here. Up here we've got part of the brain which we can call as the cerebral hemisphere, so I'm going to put C hemispheres. We've got the cerebellum, number 2, which is at the back of the brain. We have an area in the brainstem, which is the general term for 3, but more specifically 3 is actually in the pons, but essentially that is the brainstem. That there. 4 is of course the spinal cord itself. And 5 is the termination of the spinal cord, and that's known as the conus medullaris. I apologise once again because the squeak has come back with my stylus. 6 is the continuation of fibres that leave the conus medullaris. Now, the centre continuation of the conus medullaris is actually a part of the connective tissue, which is the pia, and also has a dural component. And so 6 is actually, if it was the continuation of the conus medullaris, would be known as the filum terminale. But actually, if we were to look inside the connective tissue surrounding the filum terminale, we'd find some nerve roots, and these roots would represent the posterior and anterior roots of lumbar, sacral and coccygeal nerves, passing inferiorly to reach their exit points, and together these are clustered and known as the cauda equina, which is also a term which refers to horse's tail. So we must remember that cauda equina is actually nervous tissue, while the filum terminale down here is actually an extension of the conus medullaris and the connective tissue surrounding the spinal cord. It's not actually nervous tissue itself. So that's a quick labelling of the spinal cord. So the spinal cord, we must remember, begins about at this level here, which is about the point where the foramen magnum is. And that is the point in which the spinal cord would enter through the space in the skull known as the foramen magnum in order to become the brain stem. Of course, it's the conus medullaris down here, which is where it terminates, and it terminates roughly at the level of L1 and L2, as I've mentioned in other Show Me tutorials. Before we leave this diagram, we must remember that the spinal cord is not uniform in shape, so it has 2 major swellings or enlargements, and these regions are usually associated where a collection of spinal nerves will end up leaving the spinal cord at a particular place, and there's 2 key regions where that happens. There's a cervical enlargement which occurs between C5 to T1, and you will learn about this in more detail in practical sessions to come, because this is where the brachial plexus emerges in order to supply the muscles of the upper limb. So that's between C5 and T1. And you will learn all the individual roots and trunks, etc., and individual nerves corresponding to the nerves that leave C5 to T1 as the brachial plexus in another practical that you will be having. And that's the cervical enlargement, so it's slightly larger between those regions as those nerve rootlets leave that segment or those segments. There's another one which is known as the lumbosacral enlargement, and this occurs at L1 to S3, and this is pretty similar arrangement to the brachial plexus, but this time it's called the lumbosacral plexus, and its job is to innervate the lower limbs and also structures of the inside the pelvis. So that's pretty much it for this diagram. We're going to move on and have a look at a cross section now. So now we can have a look at this diagram and have a go at labelling it. So before we talk a little bit more in detail about the precise areas in the cross section of a spinal cord or part of the spinal cord, let's just label it here. So first of all, one is essentially white matter, so keeping it very basic. So this is the white matter. This is predominantly axons that are travelling longitudinally inside the spinal cord. 2 and 3 are both grey matter, and they can be differentiated by being anterior and posterior inside the spinal cord, and that's quite important for the arrangement of nerve fibres. But essentially, if we just remind ourselves that this is actually anterior and this is posterior back here, then this number 2 is actually what we call the dorsal horn, or the dorsal area of the grey matter. Dorsal horn. And 3 would be the anterior horn of the grey matter. And this is where we'd have cell bodies of neurons, and all of the cell bodies in the dorsal horn are sensory in function, and those in the anterior horn are motor. So that's worth remembering. 6 is actually a nerve rootlet, and of course this one in particular is a nerve rootlet that is travelling to the posterior part, the dorsal horn, so they are going to be sensory nerve fibres in that rootlet. And if we actually look at the rootlets here, so we've got one here and one in here as well, we actually get, where they meet, we actually get what's known as a spinal nerve. And once we have a spinal nerve, we've actually essentially got a mixed nerve, so we've now got a piece of nervous tissue that has contained within it both sensory and motor fibres, and we then have what we can refer to as a anterior rami, or ramus, and a posterior ramus. So 5 here would be the posterior ramus, and 4 would be the anterior ramus, and that completes the labelling of this diagram. What I would like to do is to move on and draw a diagram, a cross section through a typical spinal cord. It's not a particular segment anywhere specifically. I'm just going to try and recreate that, and I just want to label some of the areas on here. So first of all, in the centre in here, so this is, again, this is going to be anterior and this is posterior. We are going to have a central canal. Okay. We are also going to have this small indentation in the front here, which is known as the anterior median fissure. So I'm just going to abbreviate that as anterior median fissure with AMF. The counterpart indentation posteriorly is known as the posterior median sulcus. So that's called the posterior median sulcus. We put PMS at the back there. I now want to take a little bit of time just to deal with the connective tissue that surrounds the spinal cord, and that has a name, a general name known as the meninges. So we'll just write up the word meninges. And there are 3 component parts to this. We have the dura, dura mater, put that in grey. I'm going to choose green to put in the next layer, which is the arachnoid. Not come out very clearly there, but the arachnoid mater. And finally, the one that is closest to the spinal cord, and I've kind of run out of colours to choose from, so I'm going to go back to red, and that is the pia mater. I'm going to use those respective colours now just to draw them on, and then we can think about where the subarachnoid space is and cerebral spinal fluid would be located. So first of all, let's take our red. We started with the pia mater, and that's actually invested very closely around, adhered to the outside surface of the spinal cord itself. And when you look at dissections, you will not really be able to tell it apart. You won't really be able to see it properly. If you look at a dissection in the laboratory, you will find that the spinal cord glistens slightly, and what you'll be looking at is the reflection of the pia mater. And sometimes you can see little small stretches of pia which are longitudinally orientated, and they are sometimes referred to as the denticulate ligaments, and these extend laterally from the cord towards the arachnoid, which is the next layer out, and often you can see these, and hopefully a demonstrator will be able to point them out. Do ask somebody to see them, because they are visible and they are evidence really of the pia's existence when looking at cadaveric specimens. The next layer, as I mentioned, is the arachnoid mater, and we're going to put that in green, so that's the next layer. Draw a line around here, and then the next layer, which we're going to do in grey outside that, is the layer of the dura, and that's the tough dura that you can see is very clear on specimens. So, as I said, the dura is a tough sheet of connective tissue, and as spinal nerves and their roots pass laterally, the dura surrounds these roots as tubular sleeves, and they merge with the outer covering of the spinal nerves that leave, and that outer covering is sometimes known in histological terms as the epineurium, and you might learn this in your histology classes. The next layer in is the arachnoid mater, which I mentioned, which is in green in this particular diagram, and it's quite a delicate thin membrane, and it is generally separated from the pia mater by the subarachnoid space, and the arachnoid mater ends at the level of vertebrae about S2. Okay. The important thing to remember is that the subarachnoid space exists between the arachnoid mater, that's the green layer here, and the pia mater, which is the red. So if we draw that in blue, it's going to occupy this space in here. So where I'm filling in this space is where we would have a space known as the subarachnoid space. So we'll just abbreviate that as sub-A space. And inside the sub-A space, we would expect to find CSF, which is cerebral spinal fluid. It's important to remember that this area in blue, the subarachnoid space, extends further inferiorly than the spinal cord. Remember that I said the spinal cord ends at around about level L1, L2. The spinal cord terminates at the conus medullaris, and the subarachnoid space extends to approximately the lower border of the vertebrae of S2, and the subarachnoid space is largest in the region inferior to the terminal end of the spinal cord, where it surrounds the cordia equina, which we saw on the other diagram. So as a consequence of that, CSF can be withdrawn from the subarachnoid space in the lower lumbar region without endangering the spinal cord. So this is really important clinically, because there is something known as a spinal tap or a lumbar puncture, and this can sometimes be carried out clinically for a number of reasons. It can be a sample can be taken to look for infection, or it can be used to inject antibiotics or other agents, and possibly sometimes anaesthesia as well. And because there is less of a risk below the level of the spinal cord, then obviously it makes sense to take a sample or to inject into the subarachnoid space and the CSF below the level of the termination of the spinal cord. Okay, well, I hope that's been useful, and that concludes everything I have to say about the spinal cord.
Subscribe to Soton Brain Hub for more videos to help explain the mysteries of the brain.